Good evening, everyone. I invite Deepashri ma'am to conduct the session.
Hello, am I audible?
Yes, you are audible, ma'am.
Okay. So last class we were dealing with Monera and we did till mycoplasma. And actually we were actually discussing with the chapter mycoplasma. And so let's just revise from mycoplasma again and then continue. Okay. So does anyone has any doubt from the mycoplasma part? What we have discussed so far, we have discussed a lot. Actually, we have read a lot. Actinomycetes, archaebacteria, halophile, and then we have started with the mycoplasma. Okay. So, can anyone tell me what was the mycoplasma? Anyone? So this is something we have already discussed in the last class. So can anyone tell me what is mycoplasma? Anyone? It is already written in the slide. So just unmute yourself and just tell anyone. So what we are reading so far. Okay, so no one? Okay, fine. So mycoplasma is also known as the PPLO, and these are, I told you that these are the simplest and the smallest free living prokaryotes. And they were when they were discovered, they were being discovered in the pleural fluids of the cattle who were being suffering from the pleuropneumonia. So pleuropneumonia is actually caused by mycoplasma, which is the PPLO. And the two scientists, Knocard and Rocks, were the first who identified pleuropneumonia. And then in the pleural fluid of the cattle, they discovered this mycoplasma. And this mycoplasma are also referred to as the molecules. And later it was found that these are the smallest and the simplest free living prokaryotes. Organisms are often called the mycoplasma-like organisms or pleuropneumonia-like organisms, and their size ranges from 0.1 to 0.15 millimeter. Okay, so these are very small, like 0.1 millimeter. So too small to be seen too small to be seen in naked eyes, but they can be seen under microscope quite clearly. Okay. Just wait for once. Okay. So coming with the next slide, we know that the plasma membrane, we all know what is a plasma membrane. What is a plasma membrane? Plasma membrane is actually the outer covering or the boundary of the cell. Okay, so this protects the cell from any kind of the damage or any kind of the harm that can be occurring to this mycoplasma. So mycoplasma forms the outer boundary of the cell, this plasma membrane, and this mycoplasma has this amount of the polysaccharide which contains acetyl glucosamine, which are associated with the cell membrane and are very much rich in cholesterols. Cell wall is totally absent, and due to the absence of their cell wall, they can change their shape and their pleuromorphic. So they can be coccoid, coccobacillus, helical, fine, branched, unbranched, and like any other prokaryotes, they possess one envelope system. Okay, so what are they are saying that they can change their shape. Okay, so since they can change their shape, and why they can change their shape? Because they have no cell wall. Remember, cell wall is just more of a thicker structure than that of the cell membrane, and that since cell wall is absent, cell membrane is present, they can just change their shape to anything. Imagine we are all humans without any bones. So obviously we could have fit in even inside the suitcase, right? So that's how the cell wall is design. So cell wall is basically the bones of the cell that is absent, and so they can occur in any shape, like coccoid, coccobacillus, then helical, fine, unbranched, branch. And like other prokaryotes, mycoplasma possess one envelope system, that is the plasma membrane. Okay. So they lack organized nucleus, endoplasmic reticulum, plastids, mitochondria, Golgi body, lysosomes, centrioles, flagella, etc. Why they lack this? Because from the very first slide, we have learned that these are prokaryotic organisms. Remember that these are prokaryotes. They are not eukaryotes. So obviously they lack this organelles like nucleus, endoplasmic reticulum, plastid, mitochondria, Golgi bodies, and so on. The genetic material is represented by a single DNA, which is naked because of the absence of the histone proteins. So when you will be learning in the further classes, you will come to know that our DNA is wrapped around histone. Why DNA is wrapped? Because the DNA is too long. So it can just cover the whole earth. So that amount of DNA is present within a cell that can't be seen within our eyes by our eyes, right? So that is the reason genetic material, that is the DNA, is always being covered with histone. But prokaryotes have no histones. Always remember this. Prokaryotes have no histones. Thus, mycoplasma have no histone. So DNA duplex is not compacted as in other prokaryotes, but instead lies coiled throughout the cytoplasm. Ribosomes are 70S, granules of various types occur here and there in the cytoplasm. Mesosomes are absent. What are mesosomes? Mesosomes are something which are the noses of bacteria. Okay, so bacteria don't have any kind of nose, but they have these special structures on their cell membrane through which they can undergo respiration. Those are the mesosomes. Enzymes lie both freely in the cytoplasm as well as associated with the plasma membrane. So this is the general organization of PPLO. They possess replicating disc at one end to assist in replication and separation of the genetic material. So since their DNA is double helical structures, circular structure, so from one cell to another cell, the DNA needs to be transferred. Okay, so when one bacteria is ready to give birth to another bacteria, two cells are dividing, and in biology dividing and multiplication means same. So two cells are multiplying, a cell is multiplying means a cell is dividing. So actually what happens, that one cell becomes two cell. So when from one cell it is becoming two cell, obviously the DNA material has to be arranged equally. So the DNA, so what do you think? The mother cell will give the total DNA to the daughter cell? No, obviously not. Then how will the mother cell will survive without the DNA? And thus nature has evolved the mechanism which is called replication of DNA. So it's just like you have notes and your friend is asking you for notes. So will you give her your notes totally? No, you will just make a Xerox copy of your notes and share it with your friends. So similarly, bacteria, while replicating, they just make two copies of the DNA. And after replication is being done, this genetic material is being separated and is being passed on to the daughter cell. Some of them live as saprophytes, but majorly live as the parasitic plants and animals. And the parasitic habit is due to the inability of most mycoplasma to synthesize the required growth factor, which is the Mycoplasma gallisepticum. Okay, so the parasitic habit is what? Due to inability of mycoplasma to synthesize growth factor. So one common example is Mycoplasma gallisepticum. They don't have any kind of the electron transport chain. I told you in the last class that for doing any kinds of the activities in our body, we need ATPs. We need a lot of ATPs. But since the electron transport chain is not available, they do produce the ATP by some other unknown mechanisms. Reproduction occurs by fission. Obviously, mycoplasma is a bacteria, and obviously, reproduction occurs by fission. Or instead of fission, sometimes branching filaments may develop that has numerous nuclear bodies. So they just get constricted or separated, and as a result, two cells are produced. And they mostly produce pleuropneumonia in domestic animals. So from that pleuropneumonia disease, Knockout and Rocks first identified this mycoplasma because they are the causative agent of the disease pleuropneumonia in animals, which is a typical pneumonia and mycoplasma urethritis in humans. Little leaf disease of brinjal and witch's broom in plants. Mycoplasmas are not affected by penicillin, which are the inhibitor of the cell wall formation, but they are inhibited by tetracycline. So actually, penicillin is an antibiotic. So different antibiotics have different modes of action. What is the meaning of the modes of the action? It simply means that all the mitochondria, all the antibiotics, some antibiotics may target mitochondria of the cell. Some antibiotics may target the cell wall of the cell. Some antibiotics some antibiotics actually target what? They target other parts of the cell. So different parts of the cells are being targeted by different antibiotics. Penicillin targets the cell wall. Okay, and we know mycoplasma has no cell wall. That is the reason they are pleomorphic. They can obtain any kind of shape. That is the reason mycoplasmas cannot be affected by penicillin. Tetracycline can inhibit mycoplasma because tetracycline targets the cell membrane. That is the reason tetracyclines can affect the mycoplasma. What are the general? So this is all about mycoplasma and pleuropneumonia-like organisms. So anything anyone wants to ask me, any question? Any doubts? Anything you can ask me regarding mycoplasma if there is something that has arisen in your mind. Is there anything? Okay, no. So what are the general characteristics of the cyanobacteria? Cyanobacteria is also known as the blue-green algae. So general characteristics are cyanobacteria are gram-negative prokaryotes which perform oxygenic photosynthesis like plants. They were the first organisms to make the atmosphere aerobic. Last class, I told you the difference between oxygenic and the anoxygenic photosynthesis. Oxygenic photosynthesis means this photosynthesis performs oxygen, like plants perform plants mostly perform photosynthesis and they give up oxygen. Okay, so during the process of the photosynthesis, they just reduce oxygen and they just reduce water molecules and produce this oxygen. So that is oxygenic photosynthesis. But there are other bacteria who can perform photosynthesis, but they don't produce oxygen. Okay, so those are the anoxygenic photosynthesis. Remember, plants need oxygen for their own respiration also. So that was the reason plants were producing oxygen through photosynthesis. Plants were not producing oxygen for us. They were producing oxygen so that they could survive. We are dependent on the plants because we can't make oxygen. So that is the reason we are dependent on the plants. But cyanobacteria plants, they can make their own oxygen and perform oxygenic photosynthesis. So they were the first organisms to make the environment aerobic. The aerobic means presence of oxygen. Blue-green algae lives virtually in all the environments containing water. And the organisms range from tropics to plains and occur in soil, freshwater, oceans. In lakes and in the ocean, they form the part of the planktons. So the blue-green algae actually lives virtually in all environment. Means they are in every part of the nature. Plains. So this part becomes important because they can ask you about blue-green algae, they can ask you about cyanobacteria, and they can ask you the place where they live, like the soil, freshwater, ocean. In lakes and in ocean, they form the part of planktons. Planktons are the types of the plants. Okay. So some cyanobacteria live in the icy water of glaciers, others in the hot spring where temperatures reach 85 degrees Celsius or more due to homopolar bonds. Some blue-green algae algae live as symbiont, while other organisms like lichens. Cyanobacteria can be unicellular, spirulina, then colonial, which is the nostoc, filamentous, which is the oscillatoria. Filaments contains one or more trichome inside a mucilage sheath. So what they are saying is that cyanobacteria, as I told you, that they are in every parts of the environment, like plains, terrace, everything. So they live even in the glaciers or even in the hot springs, less like the halophiles, where they don't depend on the temperature. So 85 degrees Celsius or more. And some blue-green algae live as the symbionts while others with different organisms. So cyanobacteria can be unicellular, colonial, filamentous. Filaments contain one or more trichome inside the mucilaginous sheath. Trichomes may be homocystous, that is without the heterocyst. I told you last class. Can anyone tell me what is a heterocyst? I told you in the last class. So please be interactive, unmute yourself and just tell me what is a heterocyst. What is a heterocyst? Please unmute yourself because I can't understand what you are understanding from the class. So just unmute yourself
Ma'am, nitrogen fixation cells.
and repeat once again.
The cells which are forming while nitrogen fixation is occurring.
Network issues.
Yes, ma'am.
I think you have some network issues. So can you please tell me what you are saying again? Unmuting yourself. Okay, so I'm just telling it to you once again, but please pay attention in class. Heterocysts are the structures which are specially found in nostoc and avena, who are the parts of the cyanobacteria. These heterocysts are thick-walled structures. Within this, nitrogen fixation occurs. Okay, within this nitrogen fixation occurs. And heterocyst is a specialized cell which is found in certain types of the cyanobacteria only, which is the Anabaena nostoc. And this cell plays an important role in nitrogen fixation, which is the process of converting atmospheric nitrogen into something that can be used by other living organisms. So these heterocysts are actually structures which are quite different from other cells like thick cell walls, lack of photosynthetic pigments, presence of enzymes, specialized membranes that prevent oxygen from entering the environment, and so on. Okay, so this time, please remember what is a heterocyst. Oscillatoria, which shows the apical oscillations. Heterocystus, which may be with heterocysts like the nostoc enabina. So I told you heterocysts are present only in a specialized category of cyanobacteria, which is the nostoc enabina. Oscillatoria is a cyanobacteria, but it has no heterocyst. Okay, nitrogen fixing nostoc and enabina lives freely as well as symbionts like Azolla, Antheroceros, Cycas roots, Gunera, and nodules of Trifolium. Right. Then the cell wall possesses an outer sheath, which is a jelly-like, slime-like, mucilaginous structure. What is this thing actually being said? Cyanobacteria has a cell wall. Mycoplasma has no cell wall. Cyanobacteria has cell wall. After completing all these parts like Monera, Protista, Fungi, Plantae, Animalia. After completing all this, I will be giving you a chart where the memory, like it's a revision note. Okay, so all the special characteristics which you can't forget are written in those revision notes. Go through that revision notes well after you have completed this detailed notes. Then it will increase your probability of being selected in the NEET from the biology part at least. So the cell wall possesses an outer sheath, which is a jelly-like slimy structure, and the cell contents are divided into two regions. Outer chromatoplasm having photosynthetic pigments and the inner colorless centroplasm. Okay, so these are basically the structures, and the cell wall is two-layered. Inner layer is the peptidoglycan or the mucopeptides, which is made up of the amino sugar and the amino acids. Amino sugar is the glucosamine. Amino acids are the muramic acid, diaminopimelic acid. In your question, just see whether they are asking you about the amino sugar or amino acid. Amino sugar is glucosamine. Amino acid is the muramic acid, diaminopimelic acid, and so on. Okay? Okay. So, cyanobacteria is what? Cyanobacteria in the cytoplasm has the photosynthetic lamella are present, and these lamella, the thylakoids, contain dominating pigments like phycocyanin, allophycocyanin, phycoerythrin, in addition to that of the chlorophyll A structures, right? So I told you that that this cyanobacteria can actually perform oxygenic photosynthesis. So to perform the oxygenic photosynthesis, they need structures. Apart from chlorophylls, what are those accessory pigments? Phycocyanin, allophycocyanin, phycoerythrin, which are the phycobilenes also. So bacteria and blue-green algae lacks the mitochondria, true vacuoles, endoplasmic reticulum. Even sterols are also absent in bacteria as well as the cyanobacteria. So just name me the accessory pigments which are present in the blue-green algae, which actually aids in the process of the photosynthesis. Just tell me. Tell me, just unmute yourself. I just now told everyone. Okay, so I don't know what you are doing all. So it's phycocyanin, allophycocyanin, and the phycoerythrin, which is the phycobilin. It's just written in the screen only. So true nucleus is actually quite absent here because I told you these are the prokaryotes, and true fibrils of DNA are either distributed throughout the cell or concentrated in the central part. Okay. So it's the chromosome that resembles the bacterial chromosome. Obviously, they are the parts of bacteria. So these chromosomes are actually the part of bacterial chromosomes. Gas vacuoles are present, which are used to regulate the buoyancy in water. Color ranges from green to deep purple to often blue-green. And these colors are produced by those accessory pigments, phycocyanin, red phycocyanin, xanthophyll. There can be blue phycocyanin and a red phycoerythrin. Okay. So is this part clear? Okay.
Excuse me, ma'am. Ma'am, your PPT is not on slideshow. Yes, now it's fine, ma'am. Thank you.
Okay, so this part we just discussed. So they have the nucleoid, which is in contact with a group of the coiled membrane called the lamellosome, and lamellosome develops from plasmid. Plasmids or the additional small DNA rings may occur. 70 ribosomes are abundant here. Cell inclusions can be alpha granules, beta granules, volutin granules, polyhedral bodies. Alpha granules are cyanophysin starch, which is similar to glycogen, and beta granules, which are the lipid droplets. So those are the volutin granules and the polyhedral granules. So the reserve food is in the form of the cyanophysin, which is the mixophysin starch. Flagellas are absent in the vegetative as well as the reproductive phase. Well-marked reproductive organs, sexual reproduction, motile reproductive bodies are also even present in cyanobacteria. And reproduction process is very simple. Because these are the bacteria, so they just reproduce by vegetative means only. And no trans of sexuality has been recorded, but gene recombination can occur through conjugation, transformation, transduction. Last class, we discussed about conjugation, transformation, transduction. Right. So can anyone tell me what is conjugation, what is transformation, what is transduction? Any one of you, just one basic statement I want to know from you all. What is this? No one? So
Ma'am,
no.
conjugation is the process through which one bacteria genetic materials transfer to other by a conjugation canal or conjugation tube.
Yeah. So very good, very good. So what is transformation and transduction? Transduction.
Transduction is a process through which virus transfers uh, their genetic material from one bacteria to other bacteria.
Yeah, very nice, very good. Okay. So next are the vegetative reproduction. So vegetative reproduction occurs by mere cell division, like those binary fission, asexual reproduction, fragmentation, those on. And heterocyst formation is characterized by the presence of thick walls and yellowish contents. And each heterocyst is made up of two walls, like an inner thin layer and the outer thick gelatinous layer. And heterocyst develops from recently dividing cells. So it may be single or it may be paired. So have you seen heterocysts? These are the thick walled structure. Do you remember that these are the thick walled structures? They are different from the other structures, and also you have to remember that these heterocysts perform nitrogen fixation. They have no oxygen within this heterocyst. Just remember these parts, okay? That will be enough for you. On germination,
Excuse me, ma'am.
yeah, say.
Ma'am, I have one doubt which is not related to this topic. Shall I ask?
Yes, you can.
Ma'am, binary fission occurs in which type of division?
Can you repeat the question?
Binary fission occurs in which type of division?
So binary fission occurs in which type of division? Like this is the question.
Yes.
Okay, so do you know what is a binary fission? Binary fission is a type of cell division. So it is actually specifically occurring in the prokaryotes. So if you ask me what binary fission occurs in which type of division, so it is a type of asexual reproduction, which is a type of cell division. Okay, so where a single
Yes, ma'am.
cell,
In cell division, that is mitotically or meiotically.
no, not that type of cell division. That is some completely different thing. So binary fission has no terms related to mitosis or meiosis. That is different concept. And this is different concept. Here just the two cells are dividing. Bas. Okay. Not the mitosis or the meiosis one. But even if you say, so but I will say that even if you want to say more, that it can be more related to mitosis. Okay, why? Because binary fission,
Okay, thank you, ma'am.
yeah. So mitosis is actually occurring in eukaryotic cell, where one cell divides into two daughter cells with same number of the chromosome as the parent cell. Okay.
Yeah.
But binary fission is a type of asexual reproduction. I already told you that they have no chromosomes like structure. Just the DNA material gets equally divided. So we can't categorize them under mitosis or meiosis. So is this doubt clear?
Okay, ma'am, thank you.
Okay. So on the germination, it gives rise to new filaments, which is the homonogia formation, and they are identified by the presence of biconcave disc or separation disc between two adjacent cells. And the filament gets broken from homonogia, and each gives rise to new filaments. Spore formation occurs like exospore, endospore, echinates, and so on. So these are the basic things. Nitrogen fixation can be shown by only specific type of bacteria. Like many bacteria, several forms of the blue-green algae have the capacity to fix atmospheric nitrogen into nitrogenous compound, and the capacity is restricted to filamentous heterocystous forms like nostoc, anabina, Olestrida, and so on. So these are the organisms that can actually perform the process of nitrogen fixation. Okay. Then under anaerobic condition, non-heterocystous conditions develop. They can also fix atmospheric nitrogen. So Glyocapsa oscillatoria. Nostoc, anabina need heterocyst for nitrogen fixation. Glyocapsa oscillatoria does not need any kind of heterocystal structures to fix the atmospheric nitrogen. Okay. Okay. So the additional capacity of nitrogen fixation along with carbon dioxide fixation makes them truly autotrophic plants. In this sense, they are considered to be largely responsible for the maintenance of the soil fertility in the tropical and the temperate region. Some species of the blue-green algae have greater contribution to increase the fertility of rice field in the tropical countries like in India. That is the reason nitrogen fixation becomes so much important for the soil fertility, because they increase the soil fertility, like Anavina, Nostoc, they increase the soil fertility. So these are the important additional aspects, like Anivina nostoc increases soil fertility by fixing atmospheric nitrogen. Spirulina is used as a source of protein-rich supplement for the diets in human. Bowls of nostoc are used as food by Chinese and South Americans. Food is called the yuko, and cyanobacteria like nostoc, cyctonym are used for reclamation of user. So which is making, so that is the other name for this sterile alkaline soil. So these are some of the economical importance of the cyanobacteria. In Sambar Lake of the Rajasthan, and Abhina, this question has already come, that what happens in the Sambar Lake of Rajasthan. So local people call it as green manure. Some species of Anabina toliferates help in the conservation of the soil. Few cyanobacteria are located inside that the lichens actually help in the plant succession. Oscillatoria is even used as a pollution indicator. So oscillatoria is increasing the fertility of the soil by fixing nitrogen, and also they are used as pollution indicators. So these are the things you have to remember. What are the harmful effects? Anabenas spoil the taste of drinking water, producing toxic effects. So they are increasing fertilities of the soil, but they are reducing the purity of water. Cyanobacteria appears on buildings during, have you seen on the bricks, white structures? Those are the cyanobacteria during rainy seasons. Like during this season, you can see if you have an isolated brick, then you can see. Skin infections may be caused by cyanobacteria like Linnbayer. So, toxin secreting cyanobacteria are mainly responsible for the water groups. These on the death emits foul smell, water gets contaminated, and cyanobacteria like Rivularia release toxins which are very much harmful for the aquatic fauna. What is the meaning of the word fauna? Anyone can tell me what is the meaning of the word fauna? Flora and fauna. Fauna means animals.
Flora, plants, fauna, and animals.
Yeah, fauna means animals. So fauna can be a normal animal, or fauna can be another types of the animals also. Okay, so this is the whole chapter for the what? Monera. So Monera is something huge. We have learned a lot of Monera, right? We have learned a lot of Monera. We have learned about bacterias, archibacterias, eubacterias, and from this we have learned about the photosynthetic autotrophs, which is the cyanobacteria, and the chemosynthetic autotrophs. So let's just summarize it. I will give you the summary notes after I have completed Monera, Protista, Fungi, Plantae, Animalia. Then I will be giving you the summary note. Okay. So we learned about, so we have learned about Linnaeus. So we have learned about the drawbacks of the two kingdom classifications. So these are the important points you can't forget. So we learned about Aristotle, who was the first person to give a scientific basis for classification. Then he classified into red blood and without red blood, but that was not very accepted. Then Linnaeus came. Linnaeus used the two kingdom classification, but obviously it had certain faults. Okay, then we have learned about the five kingdom classification, and five kingdom classification was proposed by Whittaker in the year 1969. This year and Whittaker name is important. So Monera, Protista, Fungi, Plantae, Animalia. And from there we have started reading Monera. So what is Monera? Monera is a prokaryotic. Monera has a non-cellulosic cell wall, which is the polysaccharide and amino acid. They have no nuclear membrane. Their body organization is cellular, and their mode of nutrition is what? Autotrophic and heterotrophic, right? So what are so starting from the Monera, we have learned about bacteria. Why? Because bacteria are the most common type of the organisms which are present in the kingdom Monera. Monera, these bacteria are the abundant microorganisms. Hundreds of bacteria are present in the soil, and they are present in different habitats, like literally they are present in everywhere. We have learned about the shapes of the bacteria. We have learned about the heterocyst of the bacteria. we have learned about the nutrition of bacteria. And from those nutrition, what we have learned, they may be autotroph, maybe heterotroph. Autotroph means someone who can produce their own food. Heterotrophs means they don't synthesize their own food but depend on the other organisms, like dead organisms for food. Then on the basis of this, we have learned about archaebacteria and eubacteria. We know archaebacteria means these are the most primitive type of organisms. They live where? In the very harsh conditions, thermophile, halophile, methanogens. Okay, even if you are forgetting those individual characters, try to remember the revision notes very thoroughly. If you can remember this detailed notes, I can guarantee you that you will be getting most of the questions common from the biology part. So these are living in the harshest habitats that are in the extreme salt areas like halophile, thermoacidophiles, methanogens, and they have a different cell wall structure. Last class, someone asked me what is unbranched branch. So remember that they live in the extreme conditions. That is the reason they have the ability, literally the ability to, for what? Ability? The ability to deal with that specific type of the conditions or the growth conditions. So archaebacteria has different cell wall structure for their survival, and methanogens are present in ruminant animals. We have learned about the methane gas, gobor gas, right? We have learned about all this. Next, we have learned about the bacteria, which is the eubacteria. So that is the cyanobacteria. PPLO, those comes in exam, but cyanobacteria comes more than PPLO. So give more priority to cyanobacteria to remember cyanobacteria. We have learned that eubacteria have cell wall. Cell wall is rigid. Flagellum is present. Flagella actually helps in the movement. But pili helps in the transmission of the genetic material. So both are the extensions of the bacteria, but flagella is the leg of bacteria and pili are like the sex organs of the bacteria. Okay. Understood. So those include the autotrophs, which may be photosynthetic, chemosynthetic, and the heterotrophs. Among the photosynthetic autotrophs, we have learned about cyanobacteria. We have learned they have chlorophyll A just like green plants. They are unicellular, colonial, filamentous, marine, like literally they are present everywhere. We have learned this. Their colonies have a gelatinous sheath. They can be found in polluted water bodies, but they can increase the fertility of the soil by fixing nitrogen. So these are the main points of cyanobacteria. What is the chemosynthetic autotrophs? Those are someone who oxidize inorganic substances for ATP production, like recycling the nutrients like phosphorus, nitrogens. Remember that last to last class or few classes back, we have learned a lot of reactions. Those are the chemosynthetic autotrophs. And next, the heterotrophic bacteria. What are the heterotrophic bacteria? So those are actually decomposers. We can find them in the curd. We can use them to produce the antibiotics. We can use them in fixing the nitrogen in leguminous plant, and even some causes different kinds of their diseases. So, and one thing more thing we have learned in this entire chapter is that they produce spores. Why they produce spores? Can anyone tell me? They produce spores because in the abnormal condition.
Sorry,
Say, Ifra. Say.
yes.
I can't hear you actually.
Ma'am, I didn't listen your question. Can you repeat again?
I asked you why they produce spores. So can you tell me why they produce the spores?
Because of being abnormal.
Because of being the abnormal condition. They are not abnormal. They don't have nodal conditions like us. So they are not abnormal, but they have different, like, suppose the environment is very bad, but bacteria have to survive. That is the reason they produce spores. That, okay, the environment is too harsh for me. Let's produce some spores. When some spores are being produced, then they actually reside within the spores. We have learned the detailed description of these spores, right? And from the spores, they can actually what? From those spores, they can actually develop into a new organism. Not new organism, that when the abnormal condition is being fading away, then they can just get rid of their spores and just come out of the environment, in the environment, and survive, grow, maybe cause disease. Remember that mostly pathogenic. What is pathogenic bacteria? Pathogenic bacteria means whatever pathogenic bacteria, even if the term pathogenic is used in any parts of your life, remember that pathogenic means someone who has the ability to cause disease. Okay. Some bacteria are building spores for their own survival, but most of the bacteria infect us. So they build the spores. Why? These spores are actually needed for the healthy environment, for their better development. So is this part clear?
Yes, madam.
Okay, ma'am. So this was all from my side.
Thank you, ma'am. Students, you again have a class tomorrow from 5 to 7. Do attend the class. Do not miss classes. If at all you, for some reason you miss the class, before next class, you please attend the recorded session. All right. Make sure that you attend the recorded session and continue the class. Otherwise, if you once miss the continuity, it will be very difficult for you to get back on track. All right.
Okay, sir.
And another thing, students, NEET students are not attending morning session classes. Morning session classes consist of physics and chemistry, which you learn in your inter, right? By PC means biology, that is botany, zoology, and physics and chemistry. If you miss physics and chemistry classes, we cannot do anything. You maybe have that confidence that you will listen to the recorded sessions, but it is not the case that is going to happen. So please attend the morning session classes. Okay, Ifra, Umar, Radhika, Rosia.
Yes, ma'am.
Yeah, so please attend. Please struggle. If you want to make your future bright, you have to take a step to work hard. Right?
Okay, madam.
You have done so much of effort to come to this place and listen to the free classes, right? Total in 100 students, only few so in so many students have then up to 200 students, but in that only few students have got selected, and that students are you, right? So just think of your worth.
Yes, ma'am.
When you got an opportunity, you have to utilize it. So please attend the morning session classes. It consists of purely chemistry and physics, which is very much needed to NEET students also, right?
Okay, madam.
Yes, ma'am.
So please attend from tomorrow. You have morning class from 5 to 7. Okay?
Yes, okay, ma'am.
Yes. Yes, students, again, we will meet tomorrow morning. That is from 5 to 7. I hope you 4 students are going to attend tomorrow's morning class. Otherwise, I'll just call you even in the morning at 5 o'clock. Please remember, okay? You
Okay,
want a
ma'am.
phone call or
Okay,
you will
ma'am.
directly join?
We will join.
Yes. That's the spirit. Okay?
Yes,
Okay,
ma'am.
ma'am.
Yes. If you
Okay,
want
ma'am.
any phone call to just remind you, you can just inform me
No, no need, ma'am. We'll join.
because
Yes.
whole family will get disturbed if I call you, right?
Yes, yes.
Yes. Okay, students. So bye everyone. Again, we'll
Ma'am,
meet.
it's better to make a call once to me.
Okay, sure, Ifra. I'll call you. Sure. I'll give
Okay,
you a
ma'am.
wake-up call. No issues.
Okay.
Me
Right?
too, madam.
For you also, Umar?
Yes, madam. It will be
Sure, Umar.
better.
Sure. Then I'll give you a wake-up call sharp at 5 o'clock. Okay, within few minutes, you have to just log in. Or else a 5
Okay,
minutes
ma'am.
before itself,
Okay, ma'am.
right?
Okay, ma'am.
Okay.
Sure. Yes. Yes.
Thank you,
Bye,
ma'am.
students. We will meet tomorrow morning, 5 to 7.
Bye, ma'am.
Bye.
Bye, madam.